Nervous system. Nervous system maintains coordination through the use of electrical and chemical processes. There are three aspects: sensory, motor, and integrative, which we will discuss throughout the system. The nervous system has a set of unique characteristics. First is excitability. Nervous cells can be stimulated or excited. All cells can do this, but neurons are the most efficient. Conductivity. Nervous cells respond to stimuli by producing electrical signals that can be conducted out to other neurons or to muscle or to a gland. Secretion. Nervous cells have the ability to secrete a neurotransmitter that can stimulate other cells. Neurotransmitter is a chemical which the nerve cells use to communicate with other cells. Nervous system has two parts: the central nervous system and the peripheral nervous system. The central nervous system, or CNS, includes the brain and spinal cord and functions in processing and integration. The PNS includes the nervous tissue, including the nerves in the rest of the body, and is responsible for detection and sensation, stimulates change in other systems, and are the doers. The second division is somatic versus autonomic. The somatic system is voluntary and is largely involved with skeletal muscle. The autonomic system is involuntary, is responsible for secretions, sensations, and control of cardiac and smooth muscle and glands. The big question to determine which system you have is to ask, do I or don't I have control? More tissues receive nerve fibers from both systems. The divisions are not necessarily mutually exclusive, especially the somatic and autonomic. The systems of both divisions work together to maintain control and homeostasis. Neurons are the functional cells of the nervous system. If one characteristic of the nervous cells is that they are excitable, neurons take the cake. They are extremely excitable. It is not enough that they are excited, but they have to make other cells excited too. They are like 3-year-olds. Once they are really excited, they have to tell everyone. So they are excitable and they conduct impulses. There are 3 types of neuron: sensory, motor, and integrative, or interneurons. Sensory or afferent neurons detect stimuli found in PNS and transmit them to CNS. Think of them like an inbox. Motor or efferent send signals to the muscles and glands from CNS. Motor neurons begin in the spinal cord, but most of the cells is in the PNS. They carry out going signals. Think of them like an outbox. Interneurons or association neurons are entirely in the CNS. Their function is to process, store, analyze, and retrieve information received from sensory neurons and pass on a response to the motor neurons. These cells are like some street lens. Traffic only goes one way. You can't use the same neuron to send messages in and out. Like you can't drive east on an westbound lane.
You can follow the path of neural signal. The sensory neurons detect a stimulus in your finger and send that information to the CNS. The interneurons determine that your finger needs to move and send an appropriate response to the motor neuron. The motor neuron then sends the message to the muscle that will move your finger. Neuron anatomy. These structures are typical of a motor neuron. Sensory neurons can be shaped slightly different. The neuron has an unique anatomy. The following structures are typical of a motor neuron. Sensory neurons have a slightly different shape. A soma is the cell body and is processing part of cell. This is where the normal cell stuff and functions occur. Within the soma, we will find Nissl bodies. Nissl bodies are rough endoplasmic reticulum. This is unique to the neuron. Dendrites are extensions of the soma that are used to detect stimulus. They are the receiving portion of a neuron. They are usually short and highly branched. The axon hillock is considered the trigger zone or where an impulse is generated. Once a signal is generated in the axon hillock, it is sent down the axon. The single axon is a long, thick extension used to send signals to other cells. The terminal branches are where the axon splits so it can branch out towards multiple cells. Compared to axons, they do not have myelin. We'll get to the details of myelin shortly. Finally, synaptic knobs. Synaptic knobs are the very ends of the branches and interacts with other cells. Think of your neuromuscular junction. They also contain neurotransmitters. This is a structural classification of neurons. Neuroglial cells. We have two types of cells in nervous system, neurons and neuroglial cells or glial cells. Neuroglia are helper cells in the nervous system. The first type of glial cells are oligodendrocytes. They make a myelin sheath, but only for the cells in CNS. They are octopus shaped with branches that reach out and wrap around neurons, whereas the Schwann cells in PNS only wrap around one cell. The branches of oligodendrocytes wrap around multiple neurons. Then comes ependymal cells. These cells resemble cuboidal epithelium and are found in CNS and produce cerebrospinal fluid. Microglial cells. Microglial cells wander around the CNS and remove dead nervous tissue, microorganisms, and other foreign matter. Clusters of microglia can be used to find damaged area. Astrocytes. These are the most abundant glial cells in CNS and have a star shape. They also have the most diverse functions. They provide a supportive framework, form the blood-brain barrier, supply nourishment, secrete nerve growth factors and influence signaling. Satellite cells are only found in PNS. Little is known of their actual function, but they surround neuron cell bodies. Schwann cells. Next slide. Schwann cells are a type of neuroglial cells that form myelin sheath around the neurons in the PNS. They wrap around the axon over and over again. The inner layers of the wrap are called myelin sheath. In this sheath, a substance called myelin is released. Myelin is fatty protein that insulates the axons, which provides protection for electrical signal. By doing so, myelin helps speed up the signal. This is analogous to the plastic coating around an electrical cord. The myelin prevents the electrical signal from dispersing and flowing in multiple directions and keeps it going straight down the axon. As the Schwann cells wrap around the axon, the cytoplasm and nucleus gets pushed to the outside layer. This outside layer is called neurolema. The neurolema can be used as a guide to help a cut axon grown back down its original path. It provides a regeneration tube to help the axons heal if the axons are damaged. Schwann cells are too small to coat the entire axon, which is really long, so it takes multiple cells to coat the one entire axons. The spaces between the cells are called nodes of Ranvier. They are not myelinated. As an impulse travels down the axon, the signal gets weaker the farther it travels. The nodes allow the signal to recharge. In brain or spinal cord, some regions look white and some other part they look gray. The gray matter contains mostly unmyelinated axons, whereas white matter is made up of mostly myelinated axons, which look white. Here is our Schwann cell. The first layer wrap really tightly and pack together. This is where the myelin will be produced. You can also see the neurolema with the Schwann cell nucleus. We can see in this diagram how a regeneration tube can form. You do not need to know the steps of regeneration, but be familiar with the concept as a role of the neurolema. Here is a diagram of neuroglial cells in relation to the neurons. The ones shown here are on the CNS. Nerve impulse. One of the key characteristics of neuron is the ability to send an electrical current. Before we go through the steps of how a neuron sends an impulse or an electrical current, there are a few terms and concepts we need to discuss. First is membrane potential or electrical potential. This is a difference in the concentration of charged particles across the membrane. It creates a potential for energy via current or flow of charged particles. If a cell has potential, it is called polarized. Resting membrane potential. This is when a nerve is not conducting an impulse, the nerve fiber is at rest. Two critical ions, sodium and potassium, are required to establish resting membrane potential. There are more sodium ion outside the cell than there are potassium ion inside the cell. The inside of the cell is negatively charged with respect to the outside of the cell. Resting potential or resting membrane potential is usually measured around minus 70 millivolts. A stimulus is anything that changes the resting membrane potential. It will cause the cell membrane to be leaky. This results in more positive ion moving into the cell, causing there to be a less of a difference between the inside and outside of the cell. The movement of the ions cause the potential to move closer to equilibrium. Then comes threshold potential. Threshold potential is the critical voltage required to send an impulse. The neurons receive constant stimulus, and as previously mentioned, this causes the membrane to leak. A stimulus is considered strong enough to pass on when enough ions have moved to cause a significant change in voltage. This is the threshold potential. It is usually minus 55 millivolt.
To continue with our terminology, we will discuss now depolarization. If threshold is reached, this opens more gates and allows more sodium ion to flood into the cell. The neuron becomes more and more permeable to sodium. The membrane potential shifts to a less negative value. Then is repolarization. After depolarization, the membrane becomes more permeable to potassium ion and allows it to leave the cell quickly. Repolarization returns the cell to its original state. An action potential is the full cycle of depolarization and repolarization due to sodium ion coming in and potassium ion going out in response to a really strong stimulus. A neuron can have 1,000 action potential in 1 second. This allows the nervous system to be very fast. Hyperpolarization is the opposite of depolarization and creates a greater difference between the inside and outside of the cell. It makes the membrane more negative and it becomes harder for an action potential to occur. Now that we are familiar with some of the terms, let us start to walk through the steps of an action potential. First is an adequate stimulus is applied to the neuron, causing the sodium to leak into the cell. Enough sodium leaks in that the threshold stimulus minus 55 millivolt is reached. Once threshold potential is reached, sodium channels open and sodium flows into the axon, causing the membrane to depolarize. As the depolarization occurs, more sodium channels open, more sodium flows in, and the membrane depolarizes even more. At a certain voltage, at plus 35 millivolts, the sodium channels start closing. Once plus 35 millivolts is reached, there is a reversal of polarity and repolarization begins. Potassium channels fully open and potassium rushes out of the cell. This counteracts the movement of sodium. The outside of the cell again becomes less positive and is referred to as repolarization. Repolarization is aided by sodium-potassium pumps. The pumps remove 3 sodium ions from the cell and brings in 2 potassium ions, thus returning the ions to their original location. This is an active process as the ions are moving against their concentration gradients and it requires ATP. Hyperpolarization. Hyperpolarization occurs. This is when the cell goes slightly past its resting potential of minus 70 millivolts because too many potassium ions have left the inside of the cell. Finally, it goes back to resting state with the aid of sodium-potassium pump. There are a few key points about action potentials that need to be mentioned and recalled. First, action potentials do not happen all at once along the entire axon. There is a cascade effect to action potentials. The first action potentials start at the axon hillock and then stimulate action potentials in the next region. There is a cascade effect with the action potential. One will trigger another one to happen nearby and so on down the line like a domino effect. As mentioned, action potentials start at the axon hillock. There are 350 to 500 gates per square microunit in the hillock compared to 50 to 75 elsewhere. Therefore, there is a greater chance of reaching threshold quicker here. This is an all or nothing event. Either threshold is reached or it is not. Once it starts, it is not reversible. Sodium and potassium are the key players and cause the change. Also, while sodium and potassium are flowing through the gate, the sodium-potassium pump is shuttling 3 sodium ions out and 2 potassium ions in to put the concentration of each ion back to resting. The pump requires ATP. The key voltage to keep in mind minus 70 millivolts for resting membrane potential, minus 55 millivolts for threshold potential, and plus 35 millivolts when repolarization occurs. You may need to review the steps of action potential a few times in order for it to sink in. This animation should help. This diagram represents the change in voltage as the membrane moves to the action potential. Step 1 is the local potential. We will define it shortly and leads to the threshold potential. which is step 2. Step 3 is depolarization, followed by the critical voltage of plus 35 as step 4. We then switch to repolarization in step 5. Step 6 is the slight hyperpolarization. And step 7 is the return to resting. Notice that once you hit the threshold potential, depolarization and repolarization happens very quickly. So here are some more definitions. A local potential is the short-range change in membrane potential and is reversible. If a stimulus is not strong enough, the change in potential will not start an action potential, and the membrane will go back to normal. A refractory period is a period of time when the neurons resist re-stimulation. There are two stages, an absolute and relative refractory period. The absolute refractory period is a portion of the action potential when no stimulus, regardless how strong, can trigger a new action potential to start in that location. The relative period is a segment of time when a very, very strong stimulus can cause another action potential to happen at that location. Saltatory conduction.
In this diagram, we can see the absolute and relative refractory periods. The absolute refractory covers the course of depolarization and repolarization. Regardless of how strong a stimulus is, the membrane cannot start another action potential. The relative refractory period occurs during hyperpolarization. During this time, an extremely strong stimulus can kick off another action potential. It will just take more movement of sodium to reach threshold than when a cell is at resting membrane potential.
This diagram shows how
saltatory conduction is conduction via skipping and jumping of an impulse down the axon. The impulse flowing down the axon is fast but slows down, runs out of juice, luckily at the nodes of Ranvier. The action potential is charged again.
Where it can be excited and
this diagram shows how the impulse travels down the axon as is recharged at the nodes. It is almost as if the impulse jumps from one node to another, hence the term saltatory. A synapse is a region where a neuron carries information toward another structure like a muscle or a gland. There are three components: an axon or presynaptic structure, synaptic cleft, and a postsynaptic structure, either another neuron or a different form of cell. Neurotransmitters are stored and released from vesicles in the presynaptic structures and travel across the cleft to the postsynaptic structure. This is how an impulse can be passed on from cell to cell. There are 60 different kinds of neurotransmitters in our body. A diagram of the synapse. Recall the neuromuscular junction.